Today's topic, Form 4 Chapter 3, Components of Plasma Membrane and the Movement of Substance Across the Plasma Membrane. By the end of this video, we will know components in the plasma membrane, what is a fluid mosaic model, passive versus active transport, and most importantly, movement of different substances across the plasma membrane.
Yo, what is up? My name is Davin and you're watching Davin Biology. As you already seen in the title screen, the first topic that I will share publicly on this channel is Form 4 Chapter 3, specifically the two topics you have seen. But if you're looking forward for other topics, what are you waiting for? Subscribe first, lor. Now, plasma membrane. By now, if you don't know what's a plasma membrane, it's actually also known as the cell membrane you learned previously in Form 1. The function is still the same. It's basically the door of the cell whereby it actually controls the movement of substance moving in and out of the cell. There are actually three ways whereby substance is going to go through the plasma membrane in and out of the cell. As you can see here, phospholipid bilayer is actually the main entrance, and you have two back doors, the channel and the carrier protein. Because most substances are actually going to pass through the phospholipid bilayer, but there are somehow certain substances that are unable to pass through it, so they sort of have to sneak in through the back door, which are also known as the channel and the carrier protein. Now, who are all these substances? We will go through it later in this video. So first, channel and carrier protein. Very important, you must know how to differentiate them because they are actually not the same. For the channel protein, also known as the pore, by the way, has like a channel down right at the center. Channel also known as a pathway hole along gut. So this channel, the shape is fixed. It's just one straight line. That's how you recognize it. For carrier protein, it's much more different. As you can see, there's actually a hole, so like a shape-like thingy over here, whereby it's actually called a binding site for molecules to bind to it before actually moving through it. I will actually do a separate video on that in the future. So carrier protein, by the way, also can change its shape. So you can see it's sort of like moving like that. Unlike the channel, it's fixed. Okay. So for the phospholipid bilayer, there are more things we need to know, more things we must know how to explain, especially when it comes to exam. Phospholipid bilayer. Now let me break this word down for you. Bilayer means two layers. So you can see there are two layers of this like double tail sperm structure. Phospholipid, it's its name. Right? Phosphate, phospho comes from the word phosphate. Lipid means oil, fats. So there are two layers of phospholipid, so we call it the phospholipid bilayer. Now, in exam, we must know how to label this thing. Its head and its tail have different names, and the names is going to help us understand its characteristics, which will also be asked in questions. So the phosphate actually mainly forms the heads, and the tail is mainly made up of oil, lipids. So its name of the tail and the head. Now we're going to break this word down again. So hydro, as you should know, means water. Phobic means you have phobia, you're afraid, you're scared of something. So this tail is basically afraid of water. It does not like water. Just like oil, we know oil doesn't mix well with water. Hence, you can see the tail is always hiding inside the membrane. Because water is always surrounding the cell. Inside the cell is also water. Hydrophilic is the opposite. Hydro again means water. Philic means you are attracted, you love water. So hence, if question asks you to explain the characteristics of the head and the tail of the phospholipid, then you have to say hydrophilic is attracted to water, hydrophobic repels water, means it moves away from water. Easy as that. Now, besides the three entrants, you can see in the plasma membrane you have two more smaller structures here known as the glycoprotein and the cholesterol. Glycoprotein, remember, it's always on the outer surface of the membrane. Hence, we know that whenever you see the glycoprotein, that will be the outside of the cell. Glycoprotein function, it acts as antigens on the cell, whereby antigens is usually used for cells to recognize each other. Now, antigens is something we'll learn more in the future. Cholesterol, it's always in between the phospholipid bilayer, whereby it actually acts as reinforcement to make the plasma membrane stronger and more flexible. And those are the components of the plasma membrane. Done. Moving on. Questions will always like to ask this as well, which is, what is the model given to the plasma membrane proposed by these two scientists, Sager and Nicholson? These two scientists' names are not important. All you need to know is that they give a nickname to our plasma membrane, which is known as the fluid mosaic model. Right here. There's only one name for that, one answer for that. After when you name it, the next thing the question will ask is explain. What is fluid? What is mosaic? So that will be a separate 2 marks question. Fluid basically means water, flexible like water. But you can't write that. You actually have to explain more of how is it fluid, how is it flexible like water. So no matter is it fluid or mosaic, first things first, remember you have to use proteins and phospholipids to help you answer these 2 words. As you see here. They say here, proteins and phospholipids are free to move sideways. So your proteins and the phospholipids, which is the channel carrier, and the phospholipids, they are free to move sideways. So they are constantly sliding up and down, left and right, make your plasma membrane like fluid, flexible water. So next thing, the mosaic. If you're wondering what's mosaic, mosaic is a kind of tile pattern. Okay, if you've never seen this before, I'll put it somewhere over here. Whereby this kind of tile, we usually see it in bathrooms, swimming pool, am I right? But the specific mosaic pattern in our phospholipid bilayer is actually looks something like this, whereby you can see there are big and small squares inside it. Exactly like our plasma membrane, the big squares are basically the proteins, the small squares are basically the phospholipids. And hence, these 2 big and small will give us the mosaic pattern. So we just have to say, protein built into phospholipids forms a mosaic pattern. And that's how we get the 2 marks. Fluid mosaic model, done. Next, permeability of plasma membrane. So this is where the main part of today's topic is how a substance is going to go through the plasma membrane, in or out. So first things first, you must know, plasma membrane, whenever we mention the word plasma membrane, at the same time, they are actually referring to the phospholipid bilayer, because 90% of the structure is made of these blue color dots, the phospholipid. Okay, so this structure, they say it is semi-permeable, that is its characteristics. Whereby only certain substances can pass through. So if question asks you to explain the characteristics, you have to say it's semi-permeable and only certain, means only some substance can pass through, like we mentioned just now. So who are these certain substances? They are actually mentioned in point number 2. The factors that determine the movement of these molecules across the phospholipid bilayer, the plasma membrane, are size, charge, or polarity. So we're going to look at these 3 factors, but mainly actually, we are only going to look at size and charge. So to pass through the main entrance phospholipid bilayer, you actually have to achieve 2 criteria, which is size and charge. Mainly, for size, you have to be small. For charge, as you can see here, you have to be uncharged. So if you fall in this category, then congrats, you can easily pass through the phospholipid bilayer. Uncharged molecules are the opposite of charged. So charged molecules are like the ions you learn in chemistry, the sodium, the chlorine ions, even the potassium ions, and plus, minus, all this kind of charge. Yeah, so if you are charged, sorry, you cannot pass through, and if you are large, the same. So this substance will have to try to sneak in through the back door, the 2 proteins just now. Non-polar. So polarity, there are basically 2 types. It's either polar or non-polar. So they are actually a special kind of molecule, which are actually doesn't fall in this category. But this molecule is still able to pass through the main entrance because it is non-polar. So if you're not small uncharged, you have to be non-polar. Then only you can pass through. So it's already written here, non-polar actually means lipid soluble molecules, oil, for example. But doesn't mean you are polar, you cannot pass through actually. So polarity is a bit different because non-polar can pass through, but doesn't mean you are polar, you cannot. Because most small uncharged molecules are actually polar, which are water soluble. Right? So now we know what are the criteria already. So we're going to go through here right now. Okay, now right before I tell you who are the substances that can or cannot, now we actually have to know that no matter what substance you are, you have to use different methods, also known as different transports, to actually go in or out of the membrane. So the two transports you can see here, active and passive transport. So this two transport is very easy to remember. You just remember the word active, passive. So if you think about the word active, it sounds like you have to put in more effort, more energy. Whereas compared to passive, passive means you just relax, you sleep. Hence, active transport, molecules that use active transport to go in or out of the cell requires energy in the form of ATP. So ATP, by the way, is a type of molecule that provides energy. Yeah, we'll do that in a separate video. Passive transport is the opposite, since I say rest, sleep, relax. So I do not require energy in the form of ATP. So that's one of the main thing. Number two, active transport moves from low to high concentration gradient. Passive is the opposite. It moves from high to low concentration gradient. High to low, low to high is very simple. High to low concentration gradient is just like telling you I move from more molecules to less. Whereas low to high is just the opposite. I move from less to more. Now, this triangle in the middle actually is meant to be a mountain structure. Now, it's going to help us remember passive and active more easily. So this mountain, okay, here is high, here is low. Now, imagining yourself in these two situations, going up the mountain, riding a bike, and then using the same bike, going down the mountain. So if you think about it, going up the mountain, it's always a much more difficult task because you're going against gravity. So hence, you need to put in more energy to go up the mountain. So this process is going to need ATP. So as you remember just now, if you need energy, that would be active. Okay, so hence active, you need energy because it's just like going up the mountain, low to high. Now, passive is just the opposite. So when I go down the mountain, it's a free fall, easy task. So you can just roll down. It's an effortless process. So therefore, it's something that I do not require energy. So since I do not require energy, it's passive and high to low law. So that's how I remember. So when I go down, I don't need energy. Don't need energy means it's the opposite of active, passive. So that's how we remember. So now we know the transports. Okay, let's look at the three entrants. So active, passive transport. That's our third topic. Done. The final one. How are substances going to pass through each of these entrants? So at phospholipid bilayer, hold on, I missed out one thing. Come back here. Passive transport, as you can see, there are three examples, which are osmosis, simple diffusion. You actually learned both of these in Form 3 before. Simple diffusion actually comes from the word diffusion. You just add the word simple, easy. And the third one will be a new one called facilitated diffusion. Okay, this sounds a bit more complicated, that's why it's new. Active transport only have one type, we just call it active. Okay, now we come here. Phospholipid bilayer. There are actually two types of transport that can take place here, which is the two easy one you learned previously in Form 3. If you remember, it's the osmosis and the easy diffusion. Okay, but don't say that, say simple diffusion. Alright, so if you don't know by now, osmo means water. So osmosis actually is for water. Simple diffusion, just like the diffusion you learned in your Form 3, it's for gases, oxygen and carbon dioxide. So water and gases are actually example of small, uncharged molecules. So they can pass through easily. On top of that, there's also one more molecule that uses simple diffusion, which is actually our lipid soluble molecules, the one that fall in this weird category. Alright, but we can't write lipid molecules in exam if they ask us what are the example of substance. You have to use this. So there are three types of lipid molecule substances: glycerol, fatty acid, and the fat soluble vitamin, which is vitamin, remember it as A-D-E-K. And that will be our phospholipid bilayer. Now, the two small entrants. channel and carrier. So substance that does not fulfill the criteria mentioned just now have to sneak past through the back door. So the remaining two transport now, facilitated and active. So facilitated being the more complicated one actually can occur at both of the small entrance. So I just write FD. On top of that, carrier protein also allows active transport to take place. Now let's look at the facilitated at the channel first. Facilitated at the channel protein typically allows charge molecules, charge ions to pass through. So you can see charge does not fulfill this. Okay, I fall under this category, so therefore I'm being blocked. I have to sneak past through the back door, which is now the channel protein. So example of ions again are those sodium, potassium, chlorine, those kind of charge plus minus one. Now, but the facilitated at the carrier, it's for different substances. It's for now large molecules. So again, large does not fall in the category that allows to answer, which is small size. So therefore, large molecule examples are glucose and molecules. Now remember, if the question asks for example substance, you can't write large. You have to give glucose. Sorry about that. Cannot multitask. Amino acids. Okay, the proteins, the small protein, the small sugar. Active transport at the carrier protein actually allows charged ions as well. So this leads to a very common student question. When should I know charged ions is going to use facilitated through pore or active through the carrier? So very simple. Okay, when we come to protein, the key thing that we must always look at is concentration gradient. So concentration gradient, when I see ions, now let's say outside the cell I have 5 sodium ions. Inside the cell I see got 3 sodium ions. So if I diagram the exam paper showing you that the ions is moving in 5 to 3, so that is high to low. So I go down the mountain, that is passive, so hence it's facilitated diffusion. Oh, by the way, one more thing, you cannot use the word passive transport. You have to specifically state osmosis simple or facilitated. In this case, facilitated. But if I move the opposite direction, if you see the question showing the ions going out, that's just low to high, less to more. So hence I'm going to use active transport through the carrier. And also a second very common question is, look at the facilitated diffusion. Okay, they are the same transport, but it carries different substance at different parts. So ions and large molecule you cannot mix up, because when we see facilitated at the carrier protein, it's actually only for large, not for ions. Okay, these large molecules, why I go through the carrier and not pore even though it's facilitated? Because I already said they are large. Remember, channel, the size is fixed. This thing cannot fit through. So hence, I have to come to the carrier protein, which can move. Okay, so that is basically how each of the substance can move through each of the entrance. So if you think this is a bit messy, I should summarize all of those substance, transport, and the parts in this table. So the main entrance, you can see you have gases, lipid-soluble substances that uses simple diffusion. Water uses osmosis. Pore only for charge, facilitated. Carrier also for charge, but it is for low to high only. Okay, let me repeat that again. Large molecules also can pass through carrier, and I also use facilitated. Now, let's try one example question. So a very, very common question. So you can see over here, we have different substance passing through different parts of the membrane. So first things first, you have to make sure you can identify each of the parts. So P, very simple, it's passing through the main entrance. Good right here for Soluble by the ear, PP. And this triangle thingy here is passing through, you can see, straight line, not moving. That would be the channel protein. Carrier channel protein, channel P. And the one that actually can move, have hole, that is the other one, that is the carrier protein. And the last one you can see also there is movement, there's shape, that is also definitely the carrier. So once you know the parts, it's going to make your life much more easier. So for example, question A1, name the type of transport involved in substance P and Q. So for P, I pass through main entrance, there's only 2 types of transport. The easy one we learned before, osmosis, simple diffusion. Neat. So there are 2 answers for this, either one is also acceptable. Moving on, Q. Q is passing through a carrier protein. Now, carrier protein has 2 transport, right? You have active and facilitated. Again, concentration gradient, you can see high to low definitely is not active, so the remaining one, there's only one more, facilitated diffusion. And then now they want us to give example of the substance. So facilitated diffusion through carrier, it's not ions, it's large. Remember, they cannot pass through the channel because they're too big. So large molecules, but they want example. Don't just write large, you have to give either glucose or amino acid. Last one, R. Carrier protein, facilitated or active. It's quite hard to see here because you can't really see the concentration gradient, but you can see there's ATP over here. There's only one process in this chapter that we learn uses ATP and G, which is the it's the active transport. Alright, so active transport, at here we only learn there's only one type of substance, only ions, hence, just give an example of a charged ion, the easiest one, sodium ions. And that is it from me in this video. I hope you learned something and enjoyed the video. My name is Davin. Remember, this is Davin Biology, and I'll catch you guys in the next video.